Okay, good morning, everybody. Why don't we get started? So a couple of questions, really good questions, came up this morning. One about the holidays. I mentioned there were Jewish holidays this Thursday and Friday. And just to be clear, these are only holidays if you are Jewish and if you are observant. So if you're not, then just go to lab as usual. Another good question. No lab quiz this week, this Thursday or Friday. Why? Because we're doing a review for your TAs are going to be running a review for exam 1 that will be next Wednesday. So where is this a review? It's in the lab classroom. Just go to the normal lab classroom that you're used to at the time that you used to on Thursday and Friday. Instead of having lab, they're going to run a review session. Very important, go with all your questions. Review all the questions that came up that you weren't able to ask in class, or you haven't asked me, that you haven't asked them yet. They are going to be asking you to do as much of the review as possible. And of course, they'll be there to answer any questions. But this is a really valuable opportunity to get all those pesky questions that remain answered. Anybody, please, that has any further questions about the media report and what that is? I had a really good question about what if you don't have a really clear hypothesis for your topic. You have two choices. I would recommend if there is an article that you choose that's interest to you that has a clear hypothesis, that's great. But let's say there's an article that was very, very interesting to you, may not have such a clear hypothesis. What you can do is dig a little bit deeper into the neuroscience in that article, because I know all articles that you choose will have neuroscience in it, and go and find another general hypothesis that has been addressed in that general field. And that will be your hypothesis kind of requirement for this media report. Again, feel free to ask me or any of the TAs if you're not sure whether an article is appropriate. Any questions about the lab, about the review, about where you go, about the holidays? Okay, great. Any TAs, any other things you want me to announce? Okay, Monday night, 8 p.m., Meyer, room 815, will be the alternative review for those who can't be at the lab session because of the Jewish holiday, and everybody is invited if they have a last-minute burning question. Okay, great. Why don't we get started? And here, we're going to finish up the lectures on neurotransmitters, drugs in the brain. We left you with our movie showing you the actions of nicotine, cocaine, and marijuana. Was that helpful to be able to see kind of the 3D of what drugs are doing and what receptors are doing? Was that helpful to you? Have you seen that before, or was it helpful? Helpful? Good. Okay, good. I thought that movie was really good as well. And I went over this very, very quickly, but to summarize, these are the major points that I want you to know about what nicotine does and how nicotine takes advantage of our intrinsic receptor systems. What the active substance in tobacco is nicotine. It acts on the ionotropic acetylcholine receptor to activate sodium channels, causing larger excitatory postsynaptic potentials, and a side effect that they talked about because of the addiction that comes from nicotine is the tremors, because there are a lot of these acetylcholine receptors in the caudate nucleus of the striatum that you will be modeling in next week's lab session. Not in this week's, but in next week's lab session. There is a whole fascinating neuroscience of addiction. There are people right here in New York. Eric Nestler comes to mind at Mount Sinai Medical School that has studied the molecular biology of addiction and how drugs and long-term use of drugs actually modify the genes that get activated in certain parts of our brain that causes these long-term effects that are so hard to pull yourself out of in addiction. Unfortunately, we don't have time to go into that, but I know that there are plenty of articles on addiction in the popular press. So if you're interested in that, take a look at that for your media report. Okay, summary of cocaine. The active substance is cocaine from the coca plant. And this one is very insidious, acts at the presynaptic terminals of dopamine neurons, these neurons that are so important for reward. So it's short-circuiting our reward system and just going and giving you this huge burst of, or jolt of reward by blocking reuptake pumps on the presynaptic dopaminergic terminals and thereby increasing the level of dopamine in the synaptic terminals. And as I just said, because dopamine underlies a major reward system in the brain that we talked about last week, in the first part of the lecture, you get a big high and a very, very quick high and very, very addictive outcome from even a single use of cocaine. And finally, marijuana, THC. One thing that I wanted to point out is in the movie, if you remember, they said that the endogenous transmitter that uses the THC receptor was just recently found. So here's an example of, we had this drug, lots of people were using it, people were interested in understanding the neurobiology. We identified these specific receptors for THC, but we didn't know what the endogenous substance was. And this kind of the study of the drug effects allowed us to identify a brand new subcategory of neurotransmitters. That's the other thing I want to point out. So I gave you the facts of all that we know, not all that we know, but the major principles that we know about receptors in the brain and what they do and how they work. Are they ionotropic or are they metabotropic? Do we know all the neurotransmitters that exist in the brain? Who thinks we know all the neurotransmitters that exist in the brain? Very good, you all get 100%. We do not know all the neurotransmitters, so we're still discovering. I think they said that THC is one of 100 active compounds in marijuana. All of them are acting somehow. They may be acting somehow, and we're still not sure how they do. THC is a major effect, a major way marijuana acts. But these drugs are messy. It's not like, it just goes into one place. Even the dopamine. This is one way that cocaine acts on our brain. It's doing lots of other things. Some of them we know about from our neuroscience studies, some of them we don't. And then the final, THC receptors are metabotropic. These are the slower ones, and it acts as an antagonist for the THC receptors. Now, and the last point was that a major negative side effect of marijuana is that because a lot of the THC receptors are found in the hippocampus and we're antagonizing these receptors, you are shutting down the hippocampus with use of THC. Yes, question? When the movie was made, it was several years ago, not 2010, but I would say 2007. I have to look. I didn't bring it today, but I can take a look. It's not that old, and this was a group that's very into education and trying to bring more visual aids to neuroscience. So it's a very good group. So yeah. Memory loss is a serious side effect of marijuana use. And then before we move on to today's topic, which is hormones in the brain, I wanted to go over one more drug so that you know what the immediate effects are. And it's a very commonly used substance, and that's alcohol. So the effect of alcohol on the brain is, and on your behavior, as you might be aware, the effect is biphasic, an initial stimulant phase followed by a more prolonged depressant phase. So what does alcohol do? Remember at the beginning of Monday's lecture, we talked about 3 different subcategories of GABA receptors. alcohol, we know, activates the GABA A receptor. The GABA A receptor, is GABA excitatory or inhibitory? Inhibitory. So what are you doing when you're activating an inhibitory receptor? You're depressing the brain, right? You're shutting all the activity down. So GABA A receptors is coupled to a chloride channel, and by increasing that chloride channel, chloride goes in, chloride is negative, and it increases postsynaptic inhibition. It actually hyperpolarizes the postsynaptic neuron, okay? Chronic abuse of alcohol damages nerve cells and the frontal lobes, and the frontal lobes are most affected by chronic alcohol use. And even a single binge, alcohol binge, can cause brain damage. We're going to talk about an article that studied that directly in just a second. So here is just a picture of the GABA A receptor. The chloride channel is right here. Chloride can go through. Here's 3 little chloride ions, and you have the GABA site, and alcohol will activate the site right here in the middle, and will allow this channel to open, increasing the flow of chloride into the postsynaptic membrane. So several syndromes associated with too much alcohol use. This, of course, is during gestation. Fetal alcohol syndrome is a very widely studied syndrome. Fetal alcohol syndrome is associated with distinctive changes in facial features, stunted growth, and intellectual disability, irritability, tremors, and hyperactivity. These are very common changes in your overall brain function. And most strikingly, if you look at the brains of these children with fetal alcohol syndrome, here is a normal brain. Here, as I know you all are aware of, is the corpus callosum from your studies of the sheep brain and your examination of human brain pictures. Here is the brain of an infant with fetal alcohol syndrome. They've completely lost their entire corpus callosum. What is the corpus callosum? What is that doing? Yes? Connects the 2 hemispheres. So this is a bundle of axons that are bringing information from the right to the left hemisphere and from the left to the right hemisphere. What do you think would happen if you just lost your corpus callosum? It's just not there anymore. Any guesses? Yes? Right, no communication between the 2 hemispheres. What do you think that might cause? Yes? Yes, good. That's one effect. This is, of course, is not the only effect. It's just kind of one of the most visually striking that you can see from just a simple mid-sagittal slice of the brain. But a lot of this is developmental abnormality caused by chronic alcohol use in the mother. And all of that is causing the intellectual disability, tremors, and even the hyperactivity. So this is a very bad syndrome. So let's talk about what happens. Maybe you're not fetal alcohol syndrome, but you come to abuse alcohol as an adult. This, for me, is quite scary. They've studied very carefully the effects of chronic alcohol use on overall brain size in adults. These are not fetal alcohol syndrome children. These are adults that start to abuse alcohol. Here is the volume in centimeters cubed of the anterior cortical gray matter. It's just how much of your cortex do you have. Here's the volume, nice and high, in controls, first scan and second scan. And here, way down here, is the lower volume of cortex. The alcohol is actually shrinking the amount of cortical brain mass that you have. How is it doing that? You can't tell exactly from this study, but what you see here is the cortical brain mass is decreasing in these recovering alcoholics. But you see, these recovering alcoholics, of course, are not drinking during this month. And you can see an increase coming up here. A part of what's happening is they have larger lateral ventricles. Remember those lateral ventricles? What's inside the lateral ventricles? CSF, cerebral spinal fluid. These are these kind of a little fountain of cerebral spinal fluid flowing through the middle of your brain in the lateral ventricles, through the third ventricle near your thalamus, and then down into the fourth ventricle, down towards your cerebellum. Those holes in your brain get significantly larger in the recovering alcoholics. But even those lateral ventricles are coming down a little bit after a month. But a little bit scary to see the actual size changes after long-term alcohol abuse. Yes?
Why is the control in the lateral ventricle have the size going down as well?
Why does the controls in the lateral ventricle have the size going down as well? This is a small size, and I bet if they showed you the standard error, it would not be significant. It's unclear. This might be significant. They didn't show us the error, so we can't tell. This is natural variation, so I wouldn't worry about this is pretty flat. I think everybody would agree. This is going down very slightly, probably not significant. Okay, any other questions about this? Yes? Yeah, so that's a really good question. How does smaller size of the cortical gray matter or larger lateral ventricles affect brain function? So what is the effect of alcoholism on general cognition? People are not doing as well cognitively. They have poor scores on standardized tests. It's an overall depression of your brain activity, because basically you're flooding your brain with a substance that is constantly stimulating hyperpolarization. You're not able to get the normal excitatory activity in the brain. This is going to depress everything from, math scores to reading scores to everything. And it's a general widespread effect. All GABA receptors, you are stimulating all GABA A receptors when you do that. Okay? Great. Okay, and then finally, before we move on, I just wanted to share this one article. We haven't talked about individual research articles before, but I thought this was very relevant. This is an article from 2002 when they looked at the effects of CNS damage after a 4-day binge of ethanol exposure in rats. So binge drinking, as you know, can be surprisingly common in different college settings. There are schools well known for lots of binge drinking. And so what they did is they modeled this in a rodent. And what you can get is a model in the rodent where you can get the rodent addicted to alcohol. So they will self-administer alcohol over and over, and they just do it normally. It's interesting here, you cannot get a rat to binge on its own. You don't just turn on the iPod and turn down the lights and hope that they just start, drinking for 4 days. It doesn't work that way. So to do this, they have to, they actually insert a cannula and in the stomach, and they pump in the relative amount of alcohol typically consumed in a binge for 4 days. Okay, and so they've calibrated this based on the rat's weight and everything. And what they did is they had 4 days of binge with alcohol, where alcohol is pumped in on a regular basis. They had withdrawal for 4.5 days. And they did a spatial memory task to ask, well, what is this doing? What is this binge? Even if you have 4.5 days to recover, what is it doing to your memory performance on this, in this procedure? But also, they asked, well, what actually is it doing to the health of the brain? And so first I'm going to show you the anatomical effects of binge drinking. Again, this is after a 4-day binge, after withdrawal. Instead of going into the memory task, they sacrificed the animals and looked at how healthy their brain is, even after they were able to recover for 4.5 days. And the answer is that their brain looked terrible. So this is control, and this pink stain means healthy cells. And we're seeing in the olfactory bulb, in the anterior piriform cortex, and in the perirhinal cortex that we're going to be talking about in the learning and memory section, an area very important for many forms of memory. All of these cells, kind of pinkish, these dots, are healthy cells. They're looking good. Here's what happens in the binge animal. The black stain means these cells are dying or dead. You get significant cell loss in the olfactory bulb. The olfaction for rodents, as you probably know, is much, much more important than for humans. They use it for all sorts of species-specific behaviors and for survival. Lots of cell loss in the olfactory bulb. And the anterior piriform cortex actually also processes olfactory memory. All these big black dots. Here is a close-up of basically a dying cell. The black stain means that it's dying. And here is this area critical for many forms of memory, where in layer 2 and 3 of the perirhinal cortex, you're getting a lot of cell death. This is only after 4 days of binge. So these cells are dying. Do they ever come back? No, they don't. You're right. They don't come back. 4 days of binging is killing neurons in key areas of your brain. And this is shown by this animal model. But you can imagine, this is after a single binge. None of these rats have binged before. Imagine what happens if you go on multiple binges. This is going to get worse and worse. So some negative, obviously, effects of alcohol on the brain. Okay, so that is the end of the neurotransmission and drugs in the brain. I just want to go back quickly to this analogy that I talked about, where because we brought more flavor to the types of neurotransmitters and also the types of receptors present, it was analogous to taking just a black and white kind of mugshot and creating something with more color, with more flair, perhaps with more emotion. And that's what the wide range of neurotransmitters, and we haven't discovered all the neurotransmitters in the brain, and the wide range of receptors, and we haven't discovered all the receptors. That's what that's bringing to our ability to process information. And what I want to move to is kind of the last form of flavor that we're bringing to this neural communication, and that is hormones. Okay, so now I have to switch my... Okay, great. Okay, so definition of hormones. Hormones are chemicals secreted by one cell group that travel through the bloodstream to act on targets. Everybody knows what hormones are. They've been told about raging hormones when you're a teenager. But we're going to define exactly what they are, how they work, and how they differ from the classic forms of neurotransmission that we've been talking about in the last 3 lectures. Another important term is endocrine glands. Endocrine glands are made up of endocrine cells. And these are the specific cells and groups of cells that release hormones within the body. Okay? So where are the major endocrine glands, these groups of cells that release hormones in their, throughout the body, and what are their functions? Well, as you can see in men and women, they're all over the body. We're moving outside of the brain. But there are several key brain areas, and one in particular that we'll spend a lot of time on today. A major area of endocrine function, including endocrine glands, is the hypothalamus. Lots of hormones controlled and secreted by there. Pineal gland. Who saw the pineal gland in their dissection? Great. Okay. What's unique about the pineal gland? Yes? There's only one of them in the brain. It's the only unpaired structure It is a endocrine gland involved in reproductive maturation and particularly in body rhythms, night and day cycles. Okay. The next key brain area, going down a little bit, is the pituitary gland that sits just below the hypothalamus. We're going to be talking and spending quite a bit of time on the differences and the amazing functions of the anterior pituitary gland and the posterior pituitary gland. Another major gland that you have all heard of right in the middle of your throat here is the thyroid gland, important in growth and development. Adrenal glands that sit right on top of your kidneys right here are important for stress levels as well as different kinds of metabolism, salt and carbohydrate metabolism. Your pancreas is important for sugar metabolism and also secretes hormones. There are endocrine glands in your gut as well as lots of endocrine functions of the gonads, the testes and the ovaries. Okay, so that's where they are. What is a little bit about the history of our understanding of hormones? And this shouldn't be so surprising that we found out about them quite early. In the early 1800s, castration had been used a long time, and this guy, Berthold, in the early 1800s, studied specifically for the first time, although in the book it says that he was probably inspired by somebody else, studied castration in the rooster. And what he did is they found that roosters were being castrated on a regular basis, and they noticed that a castrated rooster, sorry, let's start with a normal rooster. Here's a baby rooster, and here is an adult rooster. If left undisturbed, non-castrated, a number of very sex-specific structures and behaviors develop in this male rooster. In particular, he develops a very prominent comb, the big red thing on top of his head, and the wattle, the thing on the bottom that's nice and red and attracts all the hens. He shows the behavior of mounting the hens. He's very aggressive, and he has a lot of crowing that he does. Obviously a major behavioral trait of the rooster. But let's look at group 2. These are animals whose testes were removed during development. So as a young rooster, their testes are removed. They still look normal here. But look what happens as they develop. They have a very small comb and wattle. It does develop, but it's very, very small. This castrated rooster does not mount the hens. He's not aggressive. And there's only weak crowing that the animal does. What this guy did, everybody knew this, knew what was happening. But what this guy did is he did the experiment of taking the testes of this animal that was castrated and then re-implanting it, not back in the area of the testes, but he actually re-implanted it in the abdomen and saw what happened. So this is very significant because the testes were not innervated in the same way. They were just sitting there in the abdomen, presumably still alive, the cells are still alive, and lo and behold, this rooster that was castrated but its testes were re-implanted back in the abdomen develops just like normal. A large comb and wattle, mounts hens, he's very aggressive, and he does a lot of crowing. And his conclusion was, because the re-implanted testes in this group 3 was in an abnormal body site and disconnected from normal innervation, yet still affected development, was still able to cause normal development, Berthold reasoned that the testes release a chemical signal, which he called a hormone, that has widespread effects. So there must be something in the testes that is being released on a very widespread area, and it doesn't matter whether these testes are in the normal spot that the testes are or someplace else. As long as they're in the body, they can release this substance, and the rest of the body can benefit from it. The other important thing that this early experiment demonstrated that actually he didn't realize is that it really matters when you expose these young roosters to the hormone. If he were to wait until the rooster was older and then, for example, adult, and reimplant the testes in the abdomen, no normal development would take place. This is a time-specific effect. The young roosters have to have this critical level of hormone, and it's testosterone, that they need in order to develop normally. Implanted in a much older animal, you might affect behavior a little bit, but you will never get it to come back to normal because you need the hormone at a specific developmental time to do this. Okay? So let's talk about a few of the important general principles of actions of hormones. These are unique substances, and after we go over this, we're going to be comparing them to how neurotransmitters work. So think about, as we're talking about how hormones work, how it's different from what we've talked about of how neurotransmitters work. So first, hormones act in a gradual fashion. Again, very different from neurotransmitters. It's all or none at the synaptic cleft, or the action potentials are certainly all or one. These are very gradually acting substances in the body. Hormones act by changing the probability or intensity of a particular behavior. And the relationship between behavior and hormones is reciprocal. What does that mean? Hormones can change behaviors, but behaviors can come back and modulate the level of hormone activity. And the best example, I really like the example that they give in the book, and that is the relationship between testosterone and aggressive behavior. Let's talk about it in the context of, let's say, a baboon colony in Botswana. Okay? So you have your baboon colony in Botswana, and you have 4 or 5 males in that group. And you have a number 1 male, and you have a number 5 male. These are adult males. Number 1, he's beating up. Number 1 male, he's beating up all the other males. He is going around. He can steal their food. He can slap them around. And he has high levels of testosterone. And that high level of testosterone is fueling that more aggressive behavior that he's showing. What about poor number 5? Poor number 5 is just basically going like this. Okay? So he's trying not to get beat up. He's not strong enough to beat number 1. Maybe later, if he forms an alliance or other things happen, he can beat number 1. But he is poor, lower guy on the totem pole. He is never fighting back. And if you look at his levels of testosterone, they are much lower. Okay? So that is how the relationship between behavior and hormones is reciprocal. They are affecting each other. Hormones have multiple effects, and one behavior can be affected by several hormones. So it's complicated, kind of like the different effects that different neurotransmitters can have on a given cell. Hormones often have pulsatile secretion patterns. They secrete in bursts. And this is particularly prevalent in the hormones associated with different daytime regulation, day-night regulation. You get lots of pulsatile and regular secretions of particular kinds of hormones. Some hormones are controlled by circadian clocks. That's what I was just saying. Some of them are controlled by regular circadian clocks, and they have a regular time scale in which they are secreted. They could interact with other hormones and change their effects, so an added level of complexity there. Across species, hormone structure is similar, but functions can vary, and we're going to talk about this with respect to the very last lecture that I'm going to give in this class, called The Neurobiology of Love. And we'll be talking about different hormones, vasopressin and oxytocin, that I will introduce to you in this lecture, and how those hormones vary in function across different species, but related to the same general function. Hormones can only affect cells with a receptor protein for that hormone. So in this case, this is exactly the same as neurotransmitters. Neurotransmitters cannot have effect on a postsynaptic cell unless there's a receptor there. Similarly, hormones can only have an effect on a particular cell type if there is a receptor for that hormone on the cell. Okay. So here, now that you know a little bit about what hormones do, let's talk about how they're similar and different to the neurotransmitter and neurotransmitter systems. Neural and hormone communications have similarities and differences. They both bind to receptors and can activate second messengers, the second messenger metrabotropic type of receptor activity, which brings about changes in cellular functions. So here is a regular synaptic connection. Action potentials coming down here, neurotransmitters released and affect the receptors on the postsynaptic neuron. Okay? That's how normal synaptic transmission works. Let's compare that with two different kinds of ways that hormones work in the brain and the body. Here is endocrine communication. We have an endocrine cell that is part of an endocrine gland. Let's just pretend it's that thymus gland right in the middle of your neck. That is the endocrine gland or endocrine cell. This endocrine cell gets signals from the blood. They have receptors just like other neurons, and they can be stimulated by these molecular signals. And what they do is the endocrine cells release their hormone. But the endocrine cells are releasing their hormone not into a synaptic cleft to influence a synapse. They are releasing their hormone into the bloodstream. Again, what Berthold showed in what those testes were doing in the body cavity of the young rooster. Releasing it into the bloodstream, and then this hormone can go all over the body, wherever the blood is going, and can affect all cells that it comes in contact with where there's a receptor for that particular hormone. It could affect a cell here, 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 all over the place. So a given cell, neuron, can only affect the subset of postsynaptic neurons that it is contacting. Now, it's obviously not just contacting one. We talked about that, the fact that a single axon can affect many, many cells. But that's a whole level of magnitude different from a hormone being released into the blood and being able to affect all brain areas and body areas that are affected, that have the receptor for that hormone. Now, here's another variant of that communication scheme. Here, we don't have an endocrine cell or an endocrine gland releasing a hormone. Here, we have what's called a neuroendocrine cell. It is a modified neuron. This neuron is stimulated via synapses and EPSPs and IPSPs. And when it gets stimulated, this neuroendocrine cell, a modified neuron, will then release hormone. But this hormone is released not to another synapse, but again, into the bloodstream. And we're going to see a really amazing example of this when we get to the pituitary gland. Again, those hormones released by the neuroendocrine cell will go over and affect any target cell with a receptor for those particular hormones. Okay? So we are contrasting this that I'm going to call fast and quite specific to this and this. Both of them are significantly slower and much more widespread. This is a larger kind of communication for widespread areas of the brain that's affecting large systems, like behavioral systems. Okay. So let's go over specifically 5 ways that neural and hormonal communication differ. First, neural communication travels to precise destinations, specific synapses. Hormonal communication spreads throughout the body and is picked up by cells with the proper receptor. Okay? Widespread versus specific. Second, neural messages are rapid and hormonal messages are significantly slower. Neuromodulators can influence the activity of synaptic transmitters. So hormonal messages are an example of neuromodulators. So a hormone can come and affect synaptic transmission happening, but it's affecting in a very widespread way. The distance traveled of the neurotransmitter versus the hormone varies. The synaptic cleft is very, very small, can only be visualized using EM, electron microscopy. And in contrast, hormones may travel over a meter throughout a large part of the body and throughout the entire brain. Neural messages are digital. Sequences are all or none potentials, the action potentials that we talked about. That is the mode of communication of the neurons and synapses. Hormonal messages are analog, are graded in strength. The signal is slow, it goes up slowly, and it does not have this all or none digital kind of quality. 5, neural communication are sometimes under voluntary control, while hormones are always involuntary. You can't say, okay, I really want to be aggressive now, let's just secrete more of that testosterone. Let's get it done. That doesn't work. In contrast, I have all voluntary control of my arm. You all have voluntary control of your writing hand as you're writing down notes. That is through neuronal synaptic communication. Okay, so let's talk about the 3 major categories of hormones in the body. There are protein hormones made up of peptides or strings of amino acids. There are amine or amine hormones, modified amino acids called monoamine hormones, and steroid hormones, 4 rings of carbon atoms. Here's what they look like. There are pictures in your book. Here's a protein hormone. Here is an amine hormone that is characterized by this chemical group on it, and a steroid hormone characterized by a typical steroid group. Okay, what do hormones do exactly? Hormones exert effects on cells and tissues in the body by promoting proliferation, growth, and differentiation of cells. They also modulate cell activity. We're going to get into this more as we go into specific examples. How exactly do the 3 different types of hormones act? Protein and amine hormones act rapidly. Now let me qualify this. These are slow-acting chemicals, the hormones. So of the slowly acting hormones, these 2 are the fast ones of the slow group. Okay, this is not as fast as a neurotransmitter. But protein and amine hormones act rapidly, and when they bind to the extracellular part of the receptor changes shape, and the intracellular part then activates a second messenger. So the fast version of hormones is acting through a second messenger. You'll remember that is the slow form of synaptic transmission if you work through a second messenger system. And remember, this is not being secreted from a synaptic cleft. This is being secreted sometimes by your thymus gland, and then it has to travel all the way to your receptor in your brain someplace. So that's a very, very long time to travel. Once it gets there, it uses a slow form of a neurotransmitter, the second messenger system. But for this kind of chemical, this is the fast route of action. Protein and amine hormones, the faster one of the slow group. What's the slow one of the slow group? The slow one of the slow group is the steroid hormones. Why is it slow? It's because when steroid receptor complexes form, they alter protein production with long-lasting effects. So what's happening is steroid hormones can diffuse into the cell body. Then they bind with specific receptors, this time inside the cell. These receptors are then affecting the long-term production of proteins. This is not a fast kind of thing. It's not a receptor that's suddenly activating a channel or even a second messenger that goes and has one step more to activate channels. This goes in and affects the way that proteins are being synthesized in the DNA of individual cells. Slow, 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 but very, very powerful. Okay, So another important concept that we will focus on for hormones, but it's also very relevant for lots of other areas of neuroscience, particularly there's feedback in the neuronal transmission area, but we'll focus on this concept of feedback for hormones, is how hormones get regulated. The slow release of hormones, how does that get regulated by a process called feedback control? Hormones are regulated in a very precise way by feedback systems. These are negative feedback systems where the output feeds back and inhibits further secretion. One category of negative feedback is called an autocrine response that involves the endocrine gland releasing hormone and feeding back onto itself. So here's an example. Here's an endocrine cell. It's releasing hormone. It has a target. And if it releases too much, then it senses its own release and it slows it down. Who can come up with an example of another very common negative feedback example? Yes? Excellent. So her example was progesterone and estrogen giving negative feedback in the hypothalamus. That is a beautiful, very, very complex example of feedback. How about a simple one, not even in the brain? Yeah. Okay, insulin and glucocorticoid to control sugar levels. Perfect. That's negative feedback, too much sugar, and you get increased release of insulin. And then if there's too little sugar, then more sugar is released. Any other examples? I was just going for something really simple like the heat control in a room, where you set it at a particular level. If it gets too hot, then the heat turns off. If it gets too cold, the heat turns on. So all of these examples, all of them are negative feedback examples. Here's our simplest example of feedback, but all of these examples are beautiful. Okay. In target cell feedback, a slightly more complicated form of feedback in the cell, and we're working our way all the way up to our example for estrogen and progesterone. This is a slightly simpler example. Target cell feedback, the hormone acts on its target cells and has a biological effect. And the biological effect is detected by the endocrine gland and inhibits further release. So here's the endocrine gland. You have the hormone, you have the target cell. The target cell has some sort of response. It might be releasing something like insulin, for example. And it's the insulin that's feeding back onto the endocrine cell. So just a slightly more complicated route where the endocrine cell has to have a receptor not just for what it's producing, but for some other perhaps more complex feedback that it is detecting to be able to turn on and off and know where more and less is required. Yes?
So for the first type of feedback, the endocrine cells never get any response from the target cells. It's just seeing how much is released.
Exactly. Yes. Yeah. Very, very simple. All I care about is how much of my own substance I detect in the environment. And that can work just fine. But this is because these endocrine cells are working on behavior and actually feedback on behavior. This biological response could be a behavior like aggression, for example. So maybe too much aggression will cause a little bit of a decrease, but it will not cause it to completely shut down. But you can see that it can get very, very complicated. An even more complex endocrine system involves the brain, usually the hypothalamus. And the hypothalamus can direct hormone release. The brain detects the hormone's effect and exerts a negative feedback onto the hypothalamus. So here is the hypothalamus stimulating the endocrine cell. The endocrine cell releases hormone, and the target cell, affecting a target cell, you get your biological response that goes back and feeds back to the hypothalamus. This is just a more complicated control system. You can imagine the evolutionary forces that have derived this more complex system. Yes? all these systems vary in time as well in terms of rapid and slow? That's a very good question. The answer is yes. There are variabilities. There's not just one time scale. In general, I can say the time scale is always longer than synaptic transmission. That's pretty obvious. But within the variability of all these different hormones, I have to go all the way to the back, all of these different hormones and adrenal glands, yes, there's quite a large variability in the overall time scale that we're talking about, okay? And we're going to go over some specific examples. I'm just trying to give you the principles now, and then we'll go into a couple of key specific examples so you can see those differences. Okay, now, I'm going to give you the example of perhaps the most complicated feedback and regulatory set of actions that we know about relative to hormones, and that involves the anterior pituitary gland. That is, and in this set of actions, tropic hormones are pituitary hormones that affect other endocrine glands, and releasing hormones are used by the hypothalamus to control the pituitary release of tropic hormones. So here you have the hypothalamus. Hypothalamus gives releasing hormones. We're going to go over this, so don't worry if it's too many different words. Hypothalamus releases releasing hormones that affect the anterior pituitary. The anterior pituitary releases tropic hormones that affect, finally, we get to the endocrine cells. The endocrine cells are releasing hormones that go to the target, and the endocrine, the hormone, is giving negative feedback to both the anterior pituitary and the hypothalamus. It looks complicated. It is complicated, but it's actually a beautiful, beautiful example of this really complex system that has evolved to help us regulate very, very complicated things like growth, like sexual cycling, like milk release when new babies are born, lots of different things that get exquisitely regulated because of this complex set of interactions. Okay, so we're going to jump into the key structure here that we're going to focus on for these more complex kinds of interactions, and that is understanding the hypothalamic pituitary axis. Let me remind you where that is. This, as you all know, is the thalamus. Here's the corpus callosum. Here's your cerebellum, your pons. Right below the thalamus is the hypothalamus, and right below the hypothalamus, hanging off in a single structure. This is not exactly a brain structure, although it is unpaired, is called the pituitary gland. It is a critical gland for bodily functions, for some cognitive functions, and it is just hanging off the bottom of the hypothalamus. Here's another view where you can see kind of the brain in its skull, and here is a close-up of this sagittal view of the brain. You see the corpus callosum. Here is the form. Here is the thalamus, hypothalamus, and here's this very thin little structure hanging out, the pituitary gland. As you can see, it's protected in a casing of bone. Why? Because it's very, very important. Don't want damage to this pituitary gland to happen. Okay? That's what we're talking about. We're going to be focusing on cells that are in the hypothalamus and cells and either glands or axons that are found in the pituitary gland. Okay. Another word, terminology for pituitary gland is hypophysis. Okay? Not hypothesis, hypophysis. And it releases important hormones and has two major parts. Very important to understand. Pituitary gland has an anterior part, anterior pituitary, also called the adenohypophysis, and the posterior pituitary, called the neurohypophysis. And we'll figure out why it's called these different things. They're actually organized in very different ways. The two parts of the pituitary gland are separate in functions. The pituitary stalk, or infundibulum, connects the pituitary to the hypothalamus. So this little stalk right here is the pituitary stalk, or called the infundibulum, critical for connecting cells from the hypothalamus into the pituitary gland. The stalk contains critical blood vessels and many, many axons, which only extend into the posterior pituitary. They're not doing anything else. They're going from hypothalamus into posterior pituitary. Okay, let's take a closer look at exactly what's happening. Here's a cartoon of our hypothalamus. Here's our pituitary stalk, or infundibulum, and here is the anterior and posterior pituitary. We're going to focus first on the posterior pituitary. What we see is that there are two major nuclei in the hypothalamus. They're called the paraventricular nucleus because it's situated right next to the third ventricle, paraventricular. And the second one is called the supraoptic nucleus because it's right next to the optic chiasm right here. Can everybody picture where we are on that ventral surface of the sheep brain, where those optic chiasms, you have the optic chiasm that crosses on the bottom? Everybody saw that, right? A cranial nerve 2. Right below that in the hypothalamus is this bunch of neurons called the supraoptic nucleus. Neurons from both these two nuclei, these are neurons, they have axons. The axons are coursing through the pituitary stalk, and they terminate in the posterior pituitary. So the posterior pituitary is simply made up of the axon terminals of the neurons from the paraventricular and the supraoptic nucleus. Okay? These neurons are neuroendocrine cells. They secrete hormones. Remember? Sorry, I should have brought this thing to the, like this. These are neuroendocrine cells. They are neurons where the cell body is in the hypothalamus that secrete hormones. We're going to see how they secrete it into the bloodstream. How do they secrete it into the bloodstream? Well, turns out there is a great little set of very important capillaries that surround the posterior pituitary, that these capillaries are only there to suck up all of the hormones that these neuroendocrine cells are secreting. What are the two hormones that the neuroendocrine cells secrete? The two hormones are oxytocin and vasopressin. Okay? Oxytocin is involved in reproductive and parenting behavior, and also in uterine contraction and the milk letdown reflex in females. So what happens is when the baby suckles, there are nerve impulses, the somatosensory nerve impulses that go into the hypothalamus. And what are they stimulating? Oops. They stimulate neurons in both of these nuclei, paraventricular and supraoptic nucleus. These neurons get stimulated, and they get stimulated to fire action potentials, which causes the release not of neurotransmitter, but of oxytocin. Oxytocin then gets released, and it's sucked up by this specific capillary system that surrounds the posterior pituitary. Release of oxytocin then through this capillary system gets through the bloodstream, reaches all the way back down to the breast, and causes the release, the milk letdown reflex, that is, causes milk to be excreted from the breast. So this is a beautiful example of how hormones and behavior are reciprocally interconnected, and a much different kind of longer term and longer traveling time reflex than what we've been talking about for single neurotransmission. Okay, what else is in the posterior pituitary besides oxytocin? The other one is arginine vasopressin, or simply vasopressin. It is an antidiuretic hormone. and increases blood pressure and inhibits urine formation. So just like some nerve impulses will go to stimulate the cells in the hypothalamus to release oxytocin, other stimulations in terms of different levels of water retention is going to stimulate these cells to release vasopressin. Okay. So now we're going to move from the posterior pituitary was the easier of the two. Now we're going to end with the harder of the two, the more complicated of the two, which is what is going on in the anterior pituitary. It's more complicated because there's more steps and there's many, many more hormones being secreted by the anterior pituitary compared to the posterior pituitary. And this is where we get the addition of not just hormones, but releasing hormones and tropic hormones then go out to stimulate endocrine glands that then release the hormone. Okay? But here you can see where we are. We're still at the level of the hypothalamus going down to the anterior pituitary. Okay, so what are we looking at here? So the posterior pituitary was made up of all axons, the axons and axon terminals of neuroendocrine glands that are found in the hypothalamus. The organization of the anterior pituitary is very different. These are all cells, okay? These are cells that produce, they're endocrine cells. So the anterior pituitary is made up of endocrine cells that produce anterior pituitary hormones. So if I asked you on the test, what is the difference? What is the major difference between the anterior and the posterior pituitary? You will answer me that the posterior pituitary is made up of axon terminals, and the anterior pituitary is made up of neuroendocrine cells that produce pituitary hormones. Okay? Big major difference here. Okay, so these are cells that are producing hormones. There are no more axon terminals here. What happens and how do these anterior pituitary cells get stimulated? So we have cells in the hypothalamus that are secreting, releasing hormones. These cells release the releasing hormones into what's called hypophyseal portal veins, okay? So, arteries and veins. So this is a direction of blood flow. So this is hypophyseal blood system. And so just like the capillary system in the posterior pituitary, this capillary system is specific to be able to suck up the releasing hormones that are released by these special hypothalamus nerves, and they transport them into the anterior pituitary. What happens in the anterior pituitary? The anterior pituitary gets stimulated by the releasing hormones to release what's called a tropic hormone, a particular kind of tropic hormone. And there's yet another blood capillary system surrounding the anterior pituitary that is sucking up all of the tropic hormones that then bring them to all other parts of the body. Okay? So hypothalamic neurons synthesize releasing hormones. Axons from these cells converge on the median eminence above the pituitary stalk. So here are these neurons whose axons are converging right here. It's called the median eminence, right before the stalk starts. And it releases those hormones into this special capillary system that's only there to be able to bring these releasing hormones down just a few hundred microns away to the anterior pituitary. Okay? It's actually it's a pretty amazing feat of engineering. I mean, the brain is amazing itself, but the two things that have always fascinated me is the fact that the ventricular system exists, and there's this like little fountain spurring out cerebrospinal fluid that goes and kind of bathes your whole brain. You don't usually think about that normally. But the other amazing kind of architectural and communication design, I think, is right here in the anterior and the posterior pituitary. More impressive in the anterior pituitary, because you have specific blood vessels that are just there to bring this one signal from here to there, and another blood system to bring the tropic hormones that are released by the anterior pituitary cells into a bloodstream that will go to widespread areas of the body. Okay, so we talked about releasing hormones here. Releasing hormones act on the anterior pituitary, and the anterior pituitary are producing tropic hormones. Sorry, let me go back for a second. Releasing hormones, released by the hypothalamus, are secreted into local blood vessels called the hypophyseal portal system. You need to know that name. This is the hypophyseal portal system, bringing those releasing hormones down. And releasing hormones are carried to the anterior pituitary, which then releases the tropic hormones. The hypothalamus is influenced by circulating messages, such as other hormones, and by synaptic inputs from other brain areas. So these cells, which are the kind of starting point for all of this cascade of events that can happen, these are all influenced by all the synaptic inputs that are terminating on the hypothalamus. They can be cognitive inputs. They can be inputs from your environment. Is it dangerous out there? Is it safe enough to reproduce right now? That is what's affecting these releasing hormones to then go down and start the cascade. But what does that cascade do exactly? I told you that there were many more hormones down here. Anterior pituitary releases not 2, but 6 tropic hormones, okay? Adrenal corticotropic hormone controls the adrenal cortex and steroid hormone release. So this hormone is actually going out and controlling an endocrine gland. This happens to be in the adrenal cortex, right above the kidneys. The second anterior pituitary hormone, tropic hormone release, is called thyroid-stimulating hormone. Not so surprisingly, this tropic hormone goes and stimulates the endocrine cells in the thyroid gland. Gonadotropins influence the gonads. Follicle-stimulating hormone stimulates egg-containing follicles or sperm production. Luteinizing hormone stimulates follicles to form the corpora lutea. Prolactin stimulates lactation in females and is involved in parental behavior. Growth hormone or somatotropic hormone influences growth, mostly during sleep. And the stomach hormone ghrelin also evokes growth hormone release. So that's a lot to remember. What I want you to understand is the general principle. There's 6 different 6 different tropic hormones that are doing 6 very different things, but they're all regulated in the same way. First, by releasing hormones stimulated by neuroendocrine cells. And then you get to the anterior pituitary where a tropic hormone is released. Those tropic hormones then go to act on other endocrine glands, the adrenal cortex, the thyroid, the testes, the ovaries, mammillary glands, or they work directly on bones. Okay? So it is, again, a beautiful and complex set of regulatory mechanisms in place to get all these hormones out and to get them regulated in a particular way. I just wanted to go over in 5 minutes one second example. So one example from the posterior pituitary was the milk release effect. The second example that I'll go over from the anterior pituitary is the control of the adrenal glands. Where is that? That is here. This is releasing hormone, CRH, stimulates the tropic hormone in the anterior pituitary, ACTH, that goes down to the adrenal cortex. I told you there is, it's sitting right on top of the kidney. Adrenal glands are located on top of each kidney and secrete hormones. They are endocrine glands. In mammals, the outer 80% of the gland is the adrenal cortex, the outside, just like the neocortex. And the core 20% is called the adrenal medulla. Okay? There's actually a very complex set of interactions, and I'll have you go back and read this since this is our major example. The differences between the two parts of the adrenal gland, the cortex and the adrenal medulla. The adrenal cortex is the one I want to focus on for this particular example, and that secretes steroid hormones called adrenocorticoids. Glucocorticoids are a subgroup, and it's involved in glucose metabolism. But the one that I think is very relevant for many of the examples that we'll be talking about in the future is called cortisol. This is a glucocorticoid stress hormone that increases blood glucose and breaks down protein. Cortisol is the stress hormone, okay? So this gets released in times of stress when you need more glucose, perhaps to flee from a very, very scary situation. And it can be very, very useful in very acute bouts of stress. So I think we talked about already in this class that these stress hormones and its whole regulation from the level of the hypothalamus to the anterior pituitary to the ACTH release to stimulate the stress hormones are very important in acute stress where you have to run away from the lion, okay? You need those stress hormones because it gets glucose into your blood and it gets it to the places that it needs, that glucose is needed. It gets it to your legs to run away, okay? It takes it away. It takes blood, the same overall stress response, takes blood away from your gut because you don't need to digest right now. It stops the production of, it stops ovulation. It stops sperm production. We talked about the fact that, you know, when you're running around away from the lion, you don't have time to make more sperm. Do that later. Do your ovulation later. You need to get out right now. And so it was designed to be a chronic kind of, sorry, not a chronic, but an acute kind of response. Of course, this gets abused, and we end up with chronic stress situations. If you, for example, go to college for 4 years and you're stressed out for 4 years, what are you doing? You're stimulating this, the cortisol response because of psychological stress. And that's doing a lot of bad things because this hormone goes and affects not just your brain. In fact, there's lots of stress hormones associated with the hippocampus, and increased stress and increased cortisol and stress hormones can actually end up killing your hippocampal cells. Not only that, but you get increased blood glucose, and increased stress can be associated with long-term diseases like diabetes because you get this stress response, and what is it doing? It's increasing your blood glucose levels. So this is a very, very important one. We'll come back to this when we talk about the emotion systems in the brain, but just one example of how this complex system can be regulated.